let's go ahead and restart talking more about solute gradients. This is review, so I'm going to go quickly. Using some of the things that I tested you on or quizzed you with that clickers. I'm going to move quickly, maybe too quickly to learn, but remember in this course, most of your learning is not going to be in this room. Most of your learning is going to be working through the problems, studying, reading and thinking, rewriting your notes. Remember, for a 4-credit course in a 7-week term, we're expecting something like 20 hours a week of work. Right? That's how it comes out. Well, we're only spending 7 hours a week in this room, and you're only spending 2 hours a week in lab. So that's only 9 hours out of your 20 hours of work. And so there's, 11 or 12 hours of work. Every week is going to be necessary to understand. And if you don't understand it from this room, that's fine. You go home and you figure it out with your friends, with the book, with your notes, asking me questions. Anytime. I pride myself on responding quickly to emails. I don't respond late at night. So I'll just tell you, if you send me an email at 11 o'clock at night, I will respond the next day, well before you get up. So, if I go too fast, first, you're welcome to raise your hand and try to slow me down. But the point is, a lot of your learning is going to be done not in this room. Plan on it. Work on it. The sodium-potassium ATPase hydrolyzes ATP to ADP, and in the process pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. As a result of that, it creates gradients. And here are some typical gradients for sodium and potassium. Approximately 150 millimolar sodium outside and approximately 15 millimolar sodium inside. Do not use these concentrations on the problem set. Use the concentrations in the problem set when you solve the problem set questions. You'll see there's some variation because that's the way it is. I do not expect you to memorize these numbers or these numbers or these numbers, but you do need to know that the sodium gradient is such that sodium is high outside and low inside. Potassium is high concentration inside and low concentration outside. As you're working through the problem set, internalize those features, but not the numbers. Here is a slide that goes through the cycle of activity of the sodium-potassium ATPase. So let's get oriented. One of the things about these blobograms, turning proteins into blobs, turning lipids into blobs, one of the things about these blobograms is you got to figure out which is inside and which is outside. In this cartoon, this is inside and this is outside. That's not always true. In fact, it's atypical. Typically, the inside is down and the outside is up, but I didn't draw this picture. The ATPase, the sodium-potassium pump, the sodium-potassium ATPase, it goes by many names. It binds ATP on the inside, produced by the mitochondria. You should be thinking about glycolysis and Krebs cycle and pyruvate oxidation and electron transport chain. As soon as I say ATP, some of those things should be running in your brain. ATP binds. Sodium ions bind from the inside. The protein is in an inward-facing conformation. That is to say, it's got an opening that allows the sodium ions to bind. Okay, so look at this picture, and tell me a word that is embedded in this arrow. Somebody. Henry.
So it looks like the shift from the left side to the right side looks like conformational change of the opening.
Okay, that is one good answer. There is a conformational change. Protein conformational change has happened. That is an acceptable answer. It is not complete. There is another thing that is embedded in this arrow. Yes? Hydrolysis of ATP. In the process of hydrolysis of ATP, the enzyme is phosphorylate. Both of those things are embedded in this arrow. So when you see a blogogram like this, you turn the arrows into words. I'm going to try to do that with you for the rest of the day. Okay? Sodium, phosphate, phosphorylation, protein. What is embedded in this arrow? What are the words that describe what happens between here and here? There are several correct answers. Sarah, I see you look like you know the answer.
The enzyme conformational change lowers sodium affinity for the protein binding.
Good. Several features of conformational change. It goes from an inward-facing conformation to an outward-facing conformation. The affinity with which sodium binds is decreased. The binding site affinity, the affinity of the sodium binding sites changes, decreases the affinity with which sodium binds. Okay, so then what is embedded in this area? What's the word? One or both of those arrows. Two words. One of them begins with a B, or actually begins with a U, and the other one begins with a D. D as in David. At least in my opinion. Or maybe you could do both of them beginning with D. Maybe that was made it harder. Tell me the answers. There's two words. Anybody? You do know this, I think. I think you know this. What is sodium ions? What is the difference between sodium ions here and sodium ions here? Meg.
One of the words is unbinding.
Unbinding, or dissociation. Sodium unbinds or dissociates from those binding sites that now have lower affinity. That's a correct answer. And what's the other answer for what sodium ions are now doing in this solution? Henry?
Diffusing.
Diffusing. Diffusion is a concept that is easily forgotten, but shouldn't be. And so I kind of dwell on it, at least sometimes. Dissociation and diffusion. Okay, now there is an arrow between this conformation and this conformation. Well, I'm going to do this one. We have potassium ions that diffuse and bind to the potassium binding sites of the protein because they have high affinity for potassium in this configuration. Binding causes dephosphorylation. What's embedded in this arrow? So I think we have all the concepts. It's now just a matter of using the words. What's embedded in this arrow? Meg? Yeah, there's a big protein conformational change. decreasing the affinity for the potassium binding sites, dissociation and diffusion of potassium, binding of ATP. So I want you to practice turning cartoons into narratives or stories. It's one of the things you'll get better at in this course. I'm a firm believer that as human beings, Homo sapiens, we evolved in a society where storytelling is part of our culture. We remember stories. Now, I know these biological and biochemical and molecular modeling stories are not the most compelling stories, but they're still stories. And so turning pictures into stories is part of your learning process. Okay, this is my slide to remind you that these blobs, you know from self-phys that these blobs are not blobs. They're proteins with alpha helices and beta sheets and transmembrane domains and intracellular domains and binding sites somewhere in there, subunits, alpha, beta, I don't even know, a red subunit, a blue subunit, a gold subunit. I'm not going to dwell on proteins too much because you get a full dose of that in self-phys. But you should remember in the back of your mind, every time you see a blob like this as a protein, there's something that looks like this. And I will show you those. And I won't spend as much time in it as I just did. But that's the way it is. Okay. So now, let's talk about calcium. So there's the world's worst, simplest cartoon of a muscle cell. Okay? Now I know in your handout you have a much better cartoon of a muscle cell. So let me improve it a little bit. What is that? The self-phys level question. It's got a name. I talked to Lindsay about this, Lindsay McGee. I talked to Lindsay McGee about this yesterday. Maybe I didn't talk to her about this. You got muscle cells in self-phys, right? Did you cover contraction of muscle in self-phys this year because everything was a little different? Yeah. So what's this structure? It's got a name. It's called the T tubule. The T tubule is a penetration of the surface membrane into the interior of the cell. What is that? When you see something that looks like that, what is that? What's the name of that organelle? Mitochondria. You see something like that, that's a mitochondria. What is the name of that intracellular organelle, which is cartooned in a slightly different way in your cartoon, in a muscle cell? Does anyone remember the name of this organelle? Shout it out as I start to write the letters. Sarcoplasmic reticulum. Okay, so we have the components we need now. Oh, in your cartoon that also shows these lines at the bottom, That's the contractile, actin and myosin. That's what causes the muscle cells to contract. We'll come back to those. We're talking about solutes now. In the membrane of the sarcoplasmic reticulum is a protein. Let me use a different color. Here is a protein in the surface, in the membrane of the sarcoplasmic reticulum, and here is a similar protein in the surface membrane. Use your cell phys as I start to cartoon these proteins. Shout out their names. Nothing yet? Okay, the answer is not on your on your handout. The answer is on your slide. Right, you've seen this before. Sometimes the answer is right in front of you. What is this protein called? SERCA. Sarcoplasmic endoplasmic reticulum calcium ATPase. Sarcoplasmic endoplasmic reticulum calcium ATPase. What is this one called? PMCA. Plasma membrane calcium ATPase. It's in the plasma membrane. The surface membrane is also called the plasma membrane. Calcium ATPase. As a function of these proteins, where is calcium concentration high and where is calcium concentration low? That's hard. Where is the calcium concentration on this picture low? Olivia. Inside the cell. Is it? But this is inside the cell. Is it low here? No. No. Is it low here? Cytoplasm. Cytoplasmic calcium is low. It's in the nanomolar. Nano is 1/1,000,000 of millimolar. So it's low. The calcium concentration is high here and is high here in the millimolar range because of these calcium ATPases. There is one other protein that I need to introduce. Well, there's two other proteins that I need to introduce. Somebody shout out the name of this protein as I cartoon it. Sodium, what?
Sodium calcium exchanger?
No, not yet. We're getting there. The sodium calcium exchanger does not hydrolyze ATP. This is the What protein is that? The sodium pump, the sodium potassium ATPase. It's creating the sodium gradient that you saw in the other slide. I'm not focusing on that because we're going to get to Sarah's protein. Sarah's protein is a sodium calcium exchanger. Secondary active transport. Antiport. Taking the sodium gradient. Remember, the sodium potassium ATPase built the sodium gradient using ATP, and now the sodium gradient is being used to pump calcium out. And that's part of what keeps cytoplasmic calcium nanomolar. So we have three proteins that do low cytoplasmic calcium. Circa, PMCA, NCX. Sodium calcium exchanger. Here's the PMCA, circa, NCX. We don't even see the sodium potassium pump here, but it's got to be there in order for there to be a gradient that NCX uses. So this slide misses some of the things, and it includes some other things. We're going to talk about these channels later. We're not going to talk too much about mitochondria, but they also play a role in calcium buffering. We're going to talk about contraction later. Okay, so let's do another one of these problems. This one oh, well, we already did this. I got a little out of order with my own slides. We already did this. NCX. Low cytoplasmic calcium. Sodium pump. There's PMCA. There's NCX. We're not talking about calcium channels just yet. There's circa. So you've seen all this. I'm going to move on. Any questions about the mechanisms that maintain low nanomolar cytoplasmic calcium? So now we're moving on to some neurobiology, neurophysiology. Let me blank this. Obliest spine. Oh, I just gave you the answer. What? Okay. What is this? Oh, I got to give you some more hints. sorry about my artwork. Let's see if you can parse what I'm trying to draw. What is this? I've given you some hints.
The terminal or
Presynaptic terminal of a neuron. So this is the axon. This is trying to cartoon myelin insulation, presynaptic terminal, synaptic vesicles. What is this? Yeah. If this is the presynaptic terminal, what is this? Postsynaptic spine, or dendritic spine, postsynaptic memory. Vesicle, synaptic vesicles. What is loaded into synaptic vesicles? The name of the general name for what is loaded into presynaptic vesicles. Olivia. Neurotransmitters. So these vesicles are packed full of neurotransmitters. What are these things? These blobs that I'm drawing in the postsynaptic membrane. Now that you know that there's presynaptic neurotransmitter packaged into vesicles, what are these things? You know what it is. It begins with an R.
Receptor?
Receptors. Receptors for neurotransmitter. So we're gonna get into this in some gory detail, but you had some of this in cell phys because I taught it. I know that you did. So what is this vesicle doing right now? What does that vesicle do? What's the name of that process? Olivia? Exocytosis, or fusion of the vesicle with the plasma membrane, and neurotransmitter exocytosis or neurotransmitter release. Neurotransmitter into the synaptic cleft. Okay, and then the neurotransmitter binds to receptors and something happens in the postsynaptic cell. But where we are right now in this story is how do we get that neurotransmitter packaged back into the presynaptic terminal and packaged back into the vesicle? The recycling of neurotransmitter. Okay, so I'm gonna blow up the presynaptic terminal, making it bigger. And I'm going to make the vesicles much bigger. Okay. Tell me what protein, as soon as you know, what protein am I cartooning? What's the name of that protein? Abby? Sodium, potassium, ATPase. That's the sodium form. Building the sodium gradient in the presynaptic terminal. What do we call this protein? Generically. Some of them have very specific names. What do you suppose NT stands for? Neurotransmitter. So this is a sodium neurotransmitter co-transporter using the sodium gradient to pump neurotransmitter into the cytosol, into the cytoplasm of the presynaptic terminal. Okay? Now, let's do what the slide says I was supposed to do. Still building it up. What's the name of that protein? A proton pump, a proton ATPase. You've actually seen that. That's it's a protein that is very much like the ATP synthase in the mitochondrial membrane, which uses a proton gradient to make ATP. Here is a very similar protein using ATP to make a proton gradient. I think in self-is we told you that you can run that process both ways. In mitochondria, it's used to make ATP. In synaptic vesicles, it's used to take ATP and make a proton gradient. Very similar protein. What is this protein? A proton neurotransmitter exchanger. Using the proton gradient that was built by the proton ATPase to pump neurotransmitter from cytosol, so that was confusing, from cytosol into the vesicles. And so we have reuptake, we have taken back up neurotransmitter from the synaptic cleft into the cytosol using the sodium gradient, and taken the neurotransmitter from the cytosol into the lumen of the vesicles using the proton gradient. Okay, I'm using the concepts of primary and secondary active transport. Zoom students, I want to know, are you still with me? Somebody say something, because I'm no longer seeing a Zoom screen.
I'm here.
Good, okay, thank you. So here's the cartoon in your handout. Here is the proton pump. Here is so this is blowing up this membrane. Here is the proton gradient being built by the proton pump. And here is that proton neurotransmitter exchanger. Let's not worry about the other neurotransmitters just yet. This and this are the concepts that I'm illustrating now. What we're not seeing is the sodium neurotransmitter co-transporter here. Some of you may know that So, for example, serotonin. Serotonin is a neurotransmitter. The sodium serotonin cotransporter is the target of the SSRI class of antidepressant drugs. But we'll get to some of that when we talk about serotonin. So I'm trying to make this a little more interesting to you. I taught you about the sodium-potassium pump and the sodium neurotransmitter cotransporter, and now you know the molecular target of a class of drugs that you have either taken, or your friends are taking, or your parents are taking, or your grandparents. Somebody in this room has, it's a very commonly used drug, an antidepressant that inhibits the action of the serotonin sodium cotransporter in presynaptic terminal. I'm going to skip this because we're not going to use it too much in this course. Protein. Ion channels. I want to just talk about this for 1 minute, and then we're going to take a break again. Ion channels, membrane proteins, conformational changes from closed to open configuration. When you think of channels, you can't think of them as all closed, and you can't think of them as all open. They are undergoing conformational changes, and sometimes the stimulus to cause that conformational change is a change in voltage of the membrane. Sometimes it is binding of a ligand on the inside. Sometimes it's the binding of a ligand on the outside. The channels are undergoing conformational changes, and when the channel is open, ions flow, not always from in to out, sometimes from out to in, depending on the gradient, right? Just another statement of that. Actual protein structures, because I like not to just always do blobs. Here's a image from your textbook. We have covered the proton pump. We have covered the sodium-potassium pump. We have covered CIRCA and PMCA. We have talked about sodium neurotransmitter cotransporters. We have talked about sodium-calcium exchangers. We have talked about channels. So this is where we are going. We're going to build on this knowledge for the next 2 weeks. I'm going to leave this as homework. So I would like you to do this problem as homework. I'm going to turn off the slide. And so I'm not going to post the answers to this PowerPoint until tomorrow. I want you to work on this problem as homework. So let's take a break now, and we'll take a break now, and take a 5-minute break, stretch, move around, check your phone, do all that good stuff, and then we're going to start lecture 2 from